Good morning friends, welcome you all. My name is Dr. Ankur. I welcome you all on my channel Biomedic. Friends, in the previous lectures as we have learned about different concepts regarding photosynthesis, this time I have selected a very crisp topic of the same, that is, we will discuss about different types of photosystems, and then we will discuss about the types of photophosphorylations which takes place in the presence of light, that is the photophosphorylation. There are two types of photophosphorylation, as we all know, you guys in class 12th or which are preparing for the NEET: cyclic photophosphorylation, non-cyclic photophosphorylation. The anatomical differences of thylakoid and the different types of factors or the different types of mediators, or we can say the different types of carrier molecules which are present on the membranes of thylakoids, make it a very specific site for performing all the biochemical and photochemical reactions. So let's see, we'll start with the same. As we have discussed that when we talk about the illumination, I give you a quick recap for the same. Illumination is of two types, that is phosphorescence and fluorescence. Phosphorescence is mean the electron jumps and it takes time more than 10 to the power minus 9 seconds in the excited form, and the emission of the light rays or the emission of the energy is delayed because of any reasons, mainly because of the pigments. Fluorescence is the plants, they absorb the wavelength. Suppose they are absorbing higher wavelength, then they will emit shorter wavelength. If they are absorbing shorter wavelength, then they will emit higher wavelength. It depends. These fluorescence helps the plants to give their or to have their particular appearance. The green light, in which the rate of photosynthesis is least, because on the membrane of thylakoid all these photosystems, they are situated, they have to work accordingly. When we talk about different types of photosystems, they are also known as photoacts, photosystems, or we can say photoact. Photosystem 1, photosystem 2. Engelmann, he conducted an experiment regarding the rate of evolution of oxygen gas in experimental algae. He proposed that the oxygen loving bacteria, mean the oxygenic bacteria, they evolve oxygen absorbing the particular wavelength. As they absorb the particular wavelength, the rate of photosynthesis proceed in the same manner, and as the rate of photosynthesis proceed, mean they evolve the oxygen gas in the same fashion. When we talk about some types of bacteria which are photosynthetic bacteria having bacteria chlorophyll, this bacteria chlorophyll absorbs the sunlight and do the same mechanism as the green plants do. So Engelmann came to know that rate of photosynthesis is directly proportional to the absorption spectra, as he came to know that in that particular bacteria the oxygen evolving and the rate of the photosynthesis which was going on, the wavelength which is absorbed is similar to the absorption spectra of chlorophyll A. So by this, Engelmann came to know that the absorption spectra and the action spectra, they are the prime requisites for the rate of photosynthesis, and they are required for the rate of photosynthesis. Okay, for performing or for having a optimum rate of photosynthesis. Because as we all know that these photosystems, they are having different types of chlorophyll A molecules. Mean the photosystem 1, it is having a dimer of chlorophyll A which is different from the dimer of the chlorophyll A which is present in photosystem 2. Getting confused? No problem. Photosystem 1, the very first point, it is having a dimer of chlorophyll A which is having a maximum wavelength range which we can say to be absorbed is P700. In the reaction center is P700. Okay. In photosystem 2, the reaction center is having a dimer of a chlorophyll A which can absorb the wavelengths maximum up to P680. So these are two types of photosystems, photosystem 1, photosystem 2. When we talk about photosystem 2, so this photosystem 2 and this photosystem 1, they are having a pattern, and that pattern is known as what? The Z scheme. Mean the cyclic and the non-cyclic photophosphorylation means the formation of ATP. ATP is formed along with the proton gradient which is running and the electron transfers which takes place because we are having different types of carriers. They gain electrons, they gain protons, they transfer electrons, they transfer protons. So this stepwise or this sequential journey of proton as well as electron, it is known as what? Z scheme. Now when we talk about photosystem 2, photosystem 2, the pigment center or the reaction center is what? P680. Mean the maximum optimum wavelength range is P680. It can absorb P673 and different types of. Now this P680 is the reaction center of what? Photosystem 2. Okay, so this P680 is the reaction center of photosystem 2, and in photosystem 1, the reaction center is P700. We are having a dimer of chlorophyll A. In this also, we are having a dimer of chlorophyll A in this also. Okay, so these two systems, they are present anatomically different on the different locations, the different sites of thylakoid. Photosystem 1, it is present on non-appraised part of granal and stromal thylakoids. Mean it is present on both granal thylakoids and stromal thylakoids. Photosystem 2, it is present on the appraised part of only granal thylakoids, not on stromal thylakoids. Photosystem 1 is reduced with reduction type of process, and photosystem 2 is associated with oxidation type of process. Photosystem 1 it is involved mainly in cyclic photophosphorylation, and photosystem 2 is involved in both cyclic and non-cyclic photophosphorylation. Sorry, I just got confused. Photosystem 1 is involved in both cyclic and non-cyclic photophosphorylation, and photosystem 2 is involved in both in only cyclic. अरे क्या हो रहा है? बंद करो जरा एक मिनट, पॉज करो. Photosystem 1 is involved in both cyclic and non-cyclic photophosphorylation, and photosystem 2 is involved in only and only non-cyclic photophosphorylation. Okay, now we'll elaborate with the same. This photosystem 2 and photosystem 1, they are associated with different types of carrier molecules which help to transfer the electrons and protons to maintain the proton gradient. Okay, now coming to photosystem 2 first. Photosystem 2, it is associated with oxygen evolving complex. You are getting my point? It is mainly associated with oxygen evolving complex, which contains mainly Mn2 plus Ca2 plus Cl minus ions. In the presence of these ions, the photolysis of water takes place. The water gets photolyzed to form H plus and oxygen. Okay. Because of the photolysis of water, we are having H plus, we are having oxygen, we are having electrons, and these electrons, they excite the molecule of P680. As the molecule of P680 gets excited, the electrons of P680 molecule, chlorophyll A molecule, jumps, and they have a transition phase of 10 to the power minus 9 seconds. In between this transition phase, the energy, the electrons and the protons, they are transferred. The electrons are taken by the very first carrier molecule, that is pheophytin. Okay, so it is like that. P680, water gets photolized to form H plus electrons plus O2. Now this P680 takes these electrons, and the electrons are excited and captured by pheophytin. Okay. Here we are having Mn2 plus, Ca2 plus and Cl minus complex, that is oxygen evolving complex. Now this pheophytin, it further gets excited, same, its electron is transferred to the next stage, that is what? Plastoquinone. This plastoquinone is the site where ATP formation takes place with the help of ADP and one inorganic phosphate. The energy which is carried by the electrons when it is moving from the transition from P680 to pheophytin to plastoquinone, the energy which is transformed will help in the movement of proton along the gradient, and it will also help in the formation of ATP by the help of inorganic phosphate and ADP. All these processes, they are taking place on the membrane of thylakoid membrane, and we will be discussing this topic in chemosynthetic hypothesis of chemoosmotic hypothesis in the same chapter later on. Okay. Now this plastoquinone further transfers its electron to different types of carriers. Like for example, we are having cytochrome B6F complex. It is cytochrome B6F complex. Then it is transferred to, from cytochrome B6F complex, it is transferred to what? Plastocyanin. This plastocyanin is a copper containing metalloprotein. Okay, so from plastoquinone we are having cytochrome B6F complex, then we are having plastocyanin, and from plastocyanin it is transferred to photosystem 1 reaction center, that is P700, having a dimer of chlorophyll A molecule, okay, which is having an optimum range of absorption, P700. Now from this P700 it is transferred to a reducing protein A0, which is a chlorophyll molecule only. So this reducing protein A0 further gets excited, energized to give its electron to further quinone, quinone, okay, and this AI further gets excited to give its electron to iron sulfur protein, In higher plants we are having a combination of iron sulfur proteins like FeSa, FeSb and FeX. Okay, so we are having this combination of iron sulfur proteins. From iron sulfur proteins it is transferred to ferredoxin, Fd, ferredoxin, and from ferredoxin it is transferred to flavin proteins to be transferred further to NADP to form NADH plus. Okay, mean the protons, they are also transferred to the same reducing power, that is NADP+. Okay, so this NADP plus and along with ATP, they are the assimilatory powers which helps to assimilate the CO2 to form glucose, and this NADP plus itself, it acts as a reducing power to reduce all the materials so that it can carry the proton, and these protons which are produced in the lumen of thylakoid could be transferred to the stroma so that the stromal enzymes can process this the protons to be helpful for the formation of glucose by different types of processes. So friends, this was an overlook regarding the Z scheme. It is having a shape of Z scheme. Now, as the electrons are transferred from ferredoxin to like this NADP plus, this photophosphorylation is also related to this, and as the P700 gets excited to give its electron to A0, that is a reducing protein, then AI, then iron sulfur protein, then ferredoxin, it can be migrated towards plastoquinone. So this is what a cyclic photophosphorylation, okay, and this is what a non-cyclic photophosphorylation. So in non-cyclic photophosphorylation also the ATP formation is at the stage of plastoquinone. In cyclic photophosphorylation also the same step is having the formation of ATP, but the differences are the pigment centers are the difference, the type of pigment molecules are the difference. In photosystem 1, chlorophyll A is much more in comparison to chlorophyll B and carotenoids, but in photosystem 2 we can say carotenoids are more in comparison to the carotenoids which are present in photosystem 1, okay, because photosystem, they are having carotenoids because these carotenoids help to protect the chlorophyll molecules or the chlorophyll A and B molecules to be unquenched, to get unquenched, because they utilize or they capture the singlet oxygen or the free radical oxygen so that it cannot destroy the chlorophyll molecules. So this is the overlook regarding the Z scheme. This is non-cyclic photophosphorylation, mean P700 and P680, they works independently as well as dependently, mean we can say P700 works independently and P680 is dependent on P700. The electrons and the protons which are transferred by photosystem 2, it is a non-reversible reaction, okay, and the P700 is transferring, it is a reversible reaction, okay. Now coming to some more differences, that the location of photosystem 1 and photosystem 2. So this is cyclic photophosphorylation and this whole is non-cyclic photophosphorylation. Photophosphorylation means the formation of ATP with the help of ADP and inorganic phosphate. So this is known as what? Z scheme. Okay, the product of all these Z schemes would be, when they will be biosynthesized, they will be biocatalyzed by stromal enzymes to form what? They are going to form different types of products like carbohydrates in the presence of different types of enzymes, okay. So we will be discussing the biosynthetic phase, the dark phase in the next lectures. Till then guys, thank you. If you like the video, please subscribe it, okay. Thank you guys, thank you for watching it. Friends, please subscribe my video because I will be posting different types of videos later on this chapter, this topic and various topics of chemistry as well as biology. Okay, you can search me on Biomanic, you can search me on Kemang, you can search me Biology by Dr. Ankur Agarwal, like that. So please subscribe my videos and please press that bell icon because you will be getting the recent updates regarding all the videos. Thank you guys.